A critical initiator of many cell signaling pathways is the epidermal growth factor, or EGF, a ligand produced primarily by epithelial cells. Under normal conditions, EGF binds to the epidermal growth factor receptor on the surface of target cells, triggering a controlled cascade that regulates cell division and survival. In healthy tissues, this ligand receptor interaction is finely tuned. Cells produce EGF only when needed, and the receptors respond appropriately to maintain balance. However, in cancer cells, often establish an autocrine loop by overproducing EGF, leading to perpetual receptor activation and uncontrolled cell division, or just activates the cell receptors without any ligand that activates them. The reception phase of cell signaling is mediated by receptors such as EGFR, a receptor tyrosine kinase, or RTK. EGFR is characterized by an extracellular domain that specifically binds EGF, a single transmembrane region, and an intracellular tyrosine kinase domain. Once EGF binds the receptor dimerases, which activates its kinase functions and leads to the autophosphorylation of a specific tyrosine residues. These phosphorylated sites then act as docking stations for adapter proteins, for example GRB2 and SOS, which further relay the signal into cell. This intricate process ensures that the cell accurately interprets the signal, initiating the appropriate intracellular responses. Following receptor activation, a cascade of intracellular events is triggered. A central example is the RAS, RAF, MEK, and ERK pathway. Activated EGFR recruits adapter proteins that in turn activate the small GTPase RAS. Once activated, RAS initiates a phosphorylation cascade. It recruits and activates RAF, which phosphorylates MEK, then phosphorylates ERK. Activated ERK migrates into the nucleus, where it alters the gene expression to regulate cell proliferation and survival. Although second messengers are not the primary drivers in this cascade, they play a supporting role in modulating the signal's strength and duration. This cascade amplifies the signal and ensures that even a single ligand binding event can have a profound biological effect. So, under normal conditions, the controlled activation of these pathways result in a balanced cellular response. The regulated signal triggers precise gene expression that governs cell cycle progression, differentiation, and apoptosis. This ensures that the cell divide only when necessary, repair any damage efficiently, and die when they are no longer needed. The tight regulation of this process is essential for maintaining tissue homeostasis and overall organism health. In cancer, mutations or aberrant overexpression within these signals pathways disrupt normal cellular control. For instance, cancer cells can be overexpressed. Cancer cells can overexpress EGFR or harbor mutations in the RIS gene, resulting in the consecutive activation of the RIS, RAF, MEK, ERK cascade, even in absence of the ligand, which is EGF. This leads to continuous and unregulated cell division. Additionally, the autocrine production of growth factor creates a self-sustaining loop that further drives malignant transformation. As a result, cells ignore normal growth inhibitory signals, resist apoptosis, and initiate other cancer hallmarks such as angiogenesis and metastasis. These alterations underscore how a single disrupted signaling pathway can have widespread effect on cell behavior and overall organism health.